Hi, my name is Andrew Floeder, and I'm a fourth-year PhD student at the University of Minnesota School of Public Health. I'm excited to share my research focusing on canine oncology patients as a secondary source for antineoplastic drug exposure. In 2022, the American Cancer Society estimated that there would be more than 1.9 million new human cancer cases, an increase of over 25% in the last 15 years. Cancer is the leading cause of death in dogs, with an estimated 1 in 2 dogs developing the disease. As chemotherapy treatment increases in humans, chemotherapy for dogs is becoming more common. Chemotherapy drugs are designed to target fast-growing cells. They are unable to exclusively target cancer cells, resulting in the death of healthy normal cells. The health risks of inadvertent exposure to antineoplastic drugs are well established, with cardiac, reproductive, and genotoxic effects. Overall, this scenario may lead to the exposures of numerous patients, nurses, and family members to antineoplastic drugs. As stated, veterinary chemotherapy is increasing in oncology clinics, and the extent of this contamination is unknown. This contamination could lead towards more occupational exposures, as well as canine patients acting as a secondary source of exposure. So we decided to ask the question, what are the levels of chemotherapy contamination in veterinary oncology clinics and the patient home environment? For this approach, we used two specific aims. I'll briefly talk about the first aim, which focused on the development and optimization of a liquid chromatography selected reaction monitoring mass spectrometry method for the simultaneous identification of 6 antineoplastic drugs, before moving into the second aim, focusing on the implementation of said mass spectrometry method in oncology clinics, as well as the patient home environment for doxorubicin and cyclophosphamide. Using mass spectrometry, we identified the parent ion and fragmentation ions of all target drugs of interest. A chromatogram and spectra of cyclophosphamide is shown here as an example, with the parent mass of 261 and the two resulting fragmentation ions 140 and 120. Both fragmentation ions are used for the accurate conformation of the drug, and one is used for the quantitation. This method underwent further optimization steps with different parameters to enable a more sensitive quantitation. The sample collection and wipe method underwent optimization by spiking known amounts of the drug standard mix onto stainless steel plates to determine the recovery, limit of detection and quantitation, and method detection limits for all drugs. We conducted a field sample collection of dogs undergoing chemotherapy at the University of Minnesota Veterinary Medical Center. Wipe samples were collected in the clinic and by giving the owners a take-home kit to measure the levels of potential contamination 24 hours post-treatment, with an instructional video for the owners shown here. The sample collection consisted of the enrollment of 10 dogs wiped at 2 different time points, 5 hours in the clinic and 24 hours at home post-treatment, with either cyclophosphamide, which is an oral administered drug, or doxorubicin, an IV administered drug. The dogs were wiped using one wipe in 3 areas, the mouth, back, and hind legs, as key human contact points and potential residue for biological matrices. Additional surfaces in the kennel were collected, including the crate floor and mat. We identified cyclophosphamide on dog wipe samples exposed to the drug. Seen in the first figure, at the 5-hour time point, we were able to identify both fragment ions of cyclophosphamide and quantify the levels of the drug. This second figure shows the 24-hour time point, where we were able to identify both fragment ions, but the levels are below our limit of quantitation. We identified doxorubicin on kennel wipe samples exposed to the drug. As seen in the first figure, at the 5-hour time point, we were able to identify both fragment ions of doxorubicin and quantify the levels of the drug on the kennel floor, and the second figure shows the positive identification and quantitation of doxorubicin on the kennel mat. In the tables below are the reported levels of cyclophosphamide and doxorubicin. The cyclophosphamide detected on the canine patients ranged from 4 to 8 nanograms per sample. The doxorubicin detected on the kennel surface samples range from 18 to 1650 picograms per centimeter squared. These results may indicate that the different form of chemotherapy administration, oral or IV, may lead towards different chemotherapy contamination in different environments. It's important to note that our unique method enables for the simultaneous detection of both drugs for each sample. The dogs treated with cyclophosphamide only resulted in the identification of cyclophosphamide, and the dogs treated with doxorubicin only showed traces of doxorubicin. In conclusion, we developed a sensitive method as a new tool for the identification and quantitation of trace-level antineoplastic drug contamination. Through the implementation of said method, we conducted an exposure assessment focusing on preliminary data collected from veterinary healthcare workers administering doxorubicin and cyclophosphamide, as well as dogs or canine patients as a secondary source of exposure. This is the first study focusing on quantifying chemotherapy surface contamination in veterinary oncology clinics in combination with the potential secondary contamination in patient home environments. This research helps set the stage for surface wipe sampling to monitor all areas with potential antineoplastic drug contamination, as well as measuring different remediation strategies. It addresses the growing problem of antineoplastic drug contamination in occupational settings, as well as outside of occupational settings. This is of growing importance due to the increasing administration of home chemotherapy treatments. Furthermore, we shared all of our results with our collaborators at the Veterinary Medical Center. The research was disseminated at group meetings with the oncology technicians who may be directly impacted by the results. This prompted discussions about additional sampling, new strategies to reduce contamination, and they provided feedback in order for us to continue our successful collaboration. Thank you for listening to my presentation. Again, I would like to thank the Midwest Center for Occupational Health and Safety for funding this pilot project, both my advisors, Dr. Susan Arnold and Dr. Silvia Balbo, the Mass Spectrometry Corps, and of course, all of the oncology technicians and study participants who made my research possible. Thank you.